Thanks so much for joining us for our lesson about protein synthesis today. Protein synthesis is made up of two main steps, and we'll walk through each of these: transcription and translation. Up to this point, we've been talking a lot about DNA and how it serves as our instruction manual, how it helps make us who we are. And it does so by containing instructions for making proteins. Why do we need proteins anyway? Well, we need proteins to work as enzymes to help speed up or catalyze our chemical reactions. We also need proteins to provide our structure. Remember that proteins can serve as building blocks of cells and our bodies, like in our muscles and our skin and our hair. Some proteins also function as hormones, and some even function as antibodies or help protect us against disease. When we think about all the important jobs that proteins do, it makes sense that DNA really does determine who we are, how we look, how we work, because DNA is telling us how to build all of these proteins. Since DNA is this instruction manual to help us build our proteins, let's think about where these materials are stored and made. Keep in mind that DNA is stored in the nucleus of eukaryotic cells. But then where are proteins made? Proteins are actually made out in the cytoplasm, either free-floating ribosomes or ribosomes embedded on that rough endoplasmic reticulum. And so the big question is if DNA is stuck inside the nucleus, which it is, it cannot pass through the nuclear pores, but if proteins are made out on the ribosomes, how do the instructions in the DNA actually get communicated out to these ribosomes? And that's what we're going to figure out today. The key player that's going to help communicate these messages from the nucleus out to the ribosomes is what I like to think of as DNA's helper cousin, RNA. DNA, remember, is deoxyribonucleic acid. So its sugar is deoxyribose. On the other hand, its helper cousin, RNA, has a sugar called ribose instead. So its letters stand for ribonucleic acid. It's still made up of nucleotides. Its sugar is just a little bit different. DNA we know is double-stranded, whereas RNA is only single-stranded, as shown here in this diagram. DNA has the four bases that we're familiar with: adenine, guanine, cytosine, and thymine. RNA, on the other hand, still has adenine, guanine, and cytosine, but instead of thymine, it's got a base called uracil. So instead of T's that we see in DNA, RNA has U's. Also, DNA never leaves the nucleus. It is trapped inside, but RNA actually can leave, and this gives us that portability that we need to get the instructions from the nucleus to talk to the ribosomes out in the cytoplasm. If the goal is to take the instructions in DNA and have the cell make proteins, we're going to be starting with DNA, remember, stored in the nucleus, and through a process called transcription, DNA will be copied into an RNA form. Even though DNA is trapped in the nucleus, RNA is able to leave. It can venture out to the cytoplasm, and through a process called translation, we'll see proteins synthesized by ribosomes. In summary, transcription is the conversion of DNA to RNA, and translation is the conversion of messages in RNA being turned into amino acid chains or proteins. Let's first look at the main steps involved in transcription. Again, this is DNA being rewritten into a strand of what we call mRNA or messenger RNA, because it'll serve as a message when it leaves and goes to the cytoplasm. The first main step is that the DNA molecule must unwind and unzip. So the double helix will separate. Next, mRNA is going to enter the nucleus and rewrite a portion of one of those DNA strands. Finally, the DNA will rewind it will re-zip up into the double helix, and mRNA will be able to leave the nucleus and take that message out to the ribosome. So the DNA will unwind, RNA will be formed, and the DNA will go back to normal and the RNA will leave. As we look at that in a little more detail, the enzyme responsible for unwinding the double helix for transcription is called RNA polymerase. Its function is to help break the hydrogen bonds between the nitrogen bases. So here we can see those hydrogen bonds breaking. That's going to allow the DNA strands to separate. And in addition to unwinding the double helix, RNA polymerase is also responsible for bringing in new mRNA nucleotides, following the base pairing rules and matching up these RNA subunits with their DNA complement. Now, RNA polymerase is only going to be working off one side of this unzipped DNA molecule, and we can see that complementary bases are being brought in. C still goes with G, the T in DNA still binds with A, G still goes with C still goes with G, T still goes with A. But notice we've got a little surprise here. Adenine in DNA is going to pair up with the uracil in mRNA. So be careful when you're doing these base pairs. If at any point you think you want to write a T and you realize that it's RNA, change that to a U instead. The DNA now has unwound. New mRNA bases have been added. And now the mRNA is going to be able to break away, head out of the nucleus, and adventure off to find the ribosomes. So there goes the RNA. It's going to exit to the ribosomes. And now the DNA strand can simply reform. That double helix will re-zip. Those original base pairs will reform. And what's nice here is we can see that this blue molecule is that DNA, temporarily unwound by RNA polymerase, and new RNA bases were also added by RNA polymerase. This enzyme does it all. Complementary base pairs, and the mRNA is simply going to exit off to the side and be transported out to the cytoplasm. Again, the whole point to transcription was turning DNA into a messenger RNA copy so that message can go out and send the instructions to the ribosomes. And a simple part of this is simply our base pairing rules. So we're going to transcribe DNA into messenger RNA. Same rules as before, but remember, instead of thymines, RNA is going to have uracil instead. So who should go with this cytosine? Good, guanine. Who should go with guanine? Cytosine. T with A with, careful now, this is RNA, so we'll pair it up with uracil, C with G, A with U, G with C, C goes with G, and T still goes with A. Very good. That's all there is to transcription. Now that DNA has been converted to mRNA through the process of transcription, we're ready to head out to the ribosomes and look at the process of translation. This is where the ribosome is going to read the message from the messenger RNA and actually build a protein. The first main part of translation is that the messenger RNA leaves the nucleus and finds a ribosome. We've always known that the ribosomes were responsible for building proteins, and now we're going to see how. The ribosome reads the messenger RNA and calls over a tRNA, or a transfer RNA. The transfer RNA is responsible for bringing over the building blocks to the proteins, amino acids. And finally, as the ribosome continues to bring over tRNAs, new amino acids will be brought in, and peptide bonds will form between those amino acids. So we'll end up with a whole chain of amino acids joined together with peptide bonds, and this is a protein. We can also call it a polypeptide chain. How exactly do the ribosomes read this message? Well, the message is read in groups of 3 nitrogen bases at a time, and we call these groups of 3 codons. So for example, here's a group of 3 nitrogen bases, ACG, and that forms one codon. Here's another codon made up of 3 letters, and another codon made up of 3 letters. And it's these 3-letter groupings that the ribosomes use in order to read this message. What's nice is that as the ribosome reads these codons, it calls over a helper molecule, transfer RNA, and it knows who to call over because each codon on the messenger RNA matches with an anticodon on the tRNA. So we can see here that the codon of AUG is complementary to the anticodon of UAC. The tRNA has the anticodon, the mRNA has the codon, and since these go together, the ribosome guarantees that the right tRNA carrying the right ingredient or the right amino acid is coming over to add on to the protein chain. Let's take a look at this process in action. Here we see the ribosome, and the mRNA needs to come out and find the ribosome. How many codons are on this strand of mRNA? Codon, 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 codon. So 4 total. Once the ribosome and messenger RNA meet up, next we're going to see transfer RNA or tRNA with its anticodon and its amino acid on the other end coming in. And as the tRNA drops off the new amino acids, the ribosomes are going to help bond amino acids together using peptide bonds. Here comes the tRNA, its anticodon matching up with the codon of the mRNA. Here comes the next one, and the ribosome is going to help form that peptide bond between the adjacent amino acids. Notice that the tRNAs get out of the way, leaving amino acid joined with a peptide bond to the next amino acid with a peptide bond. And then here we see stop. And that's a great question, is how does the ribosome know where it's supposed to start reading and where it's supposed to stop? The answer to that lies in the codons. There's a specific start codon, AUG, that tells the ribosome where to begin. There are also 3 stop codons, UAA, UGA, and UAG, that tell the ribosome to stop. As the ribosome reads the mRNA, as it reads each codon, it needs to call for a specific tRNA that has the right amino acid. So the codons code for these ingredients of the proteins. And scientists know which codes bring in which amino acids, and they've created a codon chart for us. It says that if the first letter of the codon is an A, for example, we're going to start over here. If the second letter is U, we'll use this for our second letter. And if the third letter is G, we look over at the side for the third letter. We come together where the 3 of those meet up, and we see that that start codon AUG also brings in an amino acid called methionine. This can also be structured as a codon wheel. So if I want to figure out again AUG, I start with the first letter, U, G, and again I see that that results in methionine. You have a few practice codons on your notes. Go ahead and use either the wheel or the codon chart to decode and figure out which amino acids should result. We'll do more practice in class, but here are the answers just so you can double check yourself. And that concludes our look at transcription and translation.